Visual inspection of serum or plasma. Sample can identify this condition. Normal serum or plasma is light straw colored clear fluid. Hemolysis results in the sample appearing red, expressed as yellow to brown. Lipemia results in a milky or turbid appearance. Qualitative visual scales ranging from +1 to +4 indicate the relative degree of each of these conditions. Here the effect of presence of HIL, this time the absorbance, expected result, and the presence of HIL, H hemoglobin, AI ictus, L lipemia. The color or turbidity of the interferent can alter the reading taken by spectrophotometer, so the absorbance signals doesn't reflect the true concentration of analyte. This can lead to falsely high or falsely low test result. Here, the hemoglobin, here the lipemia, the ictus. Absorbance due to hemoglobin bilirubin lipemia may be measured in region utilized to spectral interpretation of analytes, and thus their absorbance can be mistaken for the dye being absorbance. Measurement at the 7 designated wavelength, the concentration of each of these interferons can be estimated. The level of HIL present can be linked with information about test method susceptibility to the interferent that was collected during evaluation of test accuracy. For example, if the test for potassium has been found to be elevated than H more than 2, but not when L or I are elevated, the laboratory may decide not to report potassium result. If H more than +2, or it can choose to report the result with conditional statement indicating the result is likely to be overestimated because of the high H index. HIL indices. Some analyzers are capable of providing a semi-quantitative estimate of HIL concentration as opposed to qualitative +1 to +4 scale using differential spectrophotometry. Assay package insert may define the HIL indexes unit and describe how they approximate the HIL concentration. The table below is an example of how the quantitative and semi-quantitative HIL indices might compare. Note that through the HIL indices may be reported with quantitative units, milligram per deciliter or gram per liter. These are only approximate estimate of concentration. Scale index of hemoglobin, ictus, lipemia, scale blank +1, +2, +3, +4, index less than 330, from 30 to 100, from 100 to 200, from 200 to 500, more than 500, lipemia, ictus, lipemia, at the ranges. A physician can interpret the test result in light of the potential effect of HIL, can request that new specimen free of interference be collected and tested. If 2 or more of the HIL interferent are present, the estimate will likely to be unreliable, and it's advisable to request that a new specimen be collected. Heterophile antibodies. Occasionally patients will have antibodies in their blood that inadvertently cross-react with antibodies in an immunoassay. The presence of these antibodies, called heterophile antibodies, can lead to falsely high or falsely low result. Common example of HAMA, human anti-mouse antibodies, which are anti-murine antibodies that develop spontaneously in some patients if exposed to mouse antigens. Patients may develop heterophile antibodies if they receive immunotherapy, a vaccine containing serum from other species, or even through environmental exposure. Sometimes the only clue that such an antibody is present is inconsistency of the test result with the patient condition. If a provider questions the result, it's possible to repeat the test adding an anti-heterophile antibody or blocking substance that will bind to the heterophile antibody in the sample before analysis. The anti-heterophile antibody or blocking substance binds to the heterophile antibody in the patient sample and prevent it from interfering in the test. Some immunoassays include anti-heterophile antibodies or blocking agent in the agent for the test to reduce the possibility of interference from heterophile antibody in the patient sample. Analyte concentration is higher than the measurement range for the test. When the analyte concentration is higher than the measurement range, also known as dynamic range, analytical measurement range, EMR, of the test, the laboratory has two options. One is to report the value as higher than the upper limit of the test method. This approach is acceptable when only knowing that the result is elevated insufficient for medical management. However, sometimes it's important to know the exact quantitative value. In such cases, the usual approach is to dilute the sample and reanalyze a diluted aliquot of the sample, mathematically correcting the measured result by dilution factor. Manual dilution and reanalysis steps are often undesirable because they are subjected to human error such as mismeasurement, miscale calculation, and use of the wrong diluent. Automated chemistry analyzer often include automatic dilution for determining the concentration of out-of-range sample without human intervention. Automated dilution, auto-dilution, when an instrument recognizes that a result is out of range, may be programmed to prepare a dilution of out-of-range sample and analyze the diluted sample. If diluted sample gives a result that is in the range, the instrument performs a calculation to correct the reported result with the dilution factor. While this process takes additional time for dilution and reanalysis, it offers the advantage of minimizing error and promptly addressing the issue and providing qualitative results from sample within minutes. No annual intervention is needed. Rate algorithm. For some enzyme rate reaction whose absorbance signals is monitored over the duration of reaction time, an alternate approach may be taken. Two different read windows, the routine reaction read windows and an earlier read windows, may be monitored to observe the reaction rate. The sample has a high concentration of analyte. The added enzymatic substrate reagent is quickly consumed, resulting in substrate depletion. The reaction no longer reflects the true enzyme quantity or activity and can't be measured. The result is reported as greater than linearity, too high to measure. In some cases, if substrate depletion is not detected as falsely low result. Here, this illustration explains the use of two read windows, a main or main routine read windows and an earlier read windows, to accurately determine the enzyme activity. For example, if less than 3 linear data points fall within the main read windows, the system will use the earlier read windows to calculate the result. The earlier read windows can use an algorithm to calculate the concentration of enzyme activity while the reaction is still in the linear range. Use of alternate read windows to extend linearity of enzyme. Random errors. Random errors are errors that occur as a result of unpredictable event that affect the measurement of the signal. Example of random errors include air bubbles, particularly matter in the sample, resulting in repeating a too small volume of sample. Random errors are unpredictable. Bubble in the sample may result in too little sample, and the result of the test will underestimate the amount of analysis. A bubble in the light path of the spectrophotometry. A bubble in the light path of spectrophotometer may cause an increase or decrease in absorbance and may lead to underestimation or overestimation of the analyte concentration. Particulate matter, often microclot, in a plasma sample that was poorly collected or inadequately mixed with anticoagulants, may impede the measurement of an aliquot for testing by clogging the sampling probe, or like bubbles by displacing some of the liquid. Microclots may be diffract light and cause error in the absorbance reading. Short sampling using less than the required volume of patient specimen in the reaction mixture cause falsely low result. Fortunately, many automated analyzers are programmed to recognize the presence of bubble, microclot, low sample volume, other random error, for example, sampling probe that measures the aliquot for analysis can sense when the sample is not following at the expected rate, and it might be if impaired by the presence of microclot and generate a pressure monitoring error for the analysis. Spectrophotometric reading are monitored with time. The instrument can recognize if the absorbance signal is not demonstrating the expected steep increase or decrease during the reaction time, and is instead showing some random high or low value, as would be seen with bubble or particle floating through the light path. When bubble or clot or other random event lead to unexpected sampling or signal pattern, the instrument can alert the operator that is the test result is suspect and need to be retested.